So in this lecture, we will study the catalytic nucleic acid. The name catalytic nucleic acid includes three words or synonyms words: DNA enzymes, ribozymes, and it is related with the nucleic acid. Therefore, RNA and DNA molecules are involved. And these nucleic acid name itself catalytic nucleic acid. It means it possesses the enzyme activities. Catalytic activities. They are capable of catalyzing specific chemical reaction that is related to the nucleic acid, as we have seen in the protein enzyme functions. So this is the nucleic acid or catalytic nucleic acids. The name ribozymes. It was first discovered in 1980s, and these scientists also given the Nobel Prize in chemistry. That I will teach you later on. So long belief that up to this we have understand that all enzymes are protein, but all proteins are not enzymes. Similarly, these all enzymes are protein. These beliefs was previously before 1980s. After that, this breakthrough to open up new possibilities or new pathways to understand the role of nucleic acid in the various cellular processes. Nowadays, in plant molecular biology and biotechnology also, they have given a tremendous use of these ribozyme or DNA enzymes. So, how to write these catalytic nucleic acids or catalytic functions of nucleic acids, or it acts as enzymes. So first you have to write discover or history behind that. There are two scientists, one is Thomas and Sydney. This Thomas Sek and Sydney Altman, they have discovered the RNA molecules that can catalyze the own splicing reaction in the thermophila, these bacteria and E. coli respectively. This has also these two scientists earned a Nobel Prize in the chemistry in 1989. So this discovery they have started in 1982. So discovery of ribozyme challenges the traditional view and the protein were the sole catalyst in the biological system. After that, they have discovered that RNA molecules could catalyze our own splicing reactions. Now, another point you have to write structure. As you know that nucleic acid having a three-dimensional structure. So catalytic nucleic acid can adopt a complex three-dimensional structure. And then they have a specific function to perform. So specific enzyme function, ribozymes typically fold. What is the functions of ribozyme? Ribozyme typically fold into intricate tertiary structure stabilized by the base pairing and non-canonical interactions. So ribozyme fold into a intricate tertiary structure that stabilized by the base pairing and non-canonical interaction. Now, catalytic DNA, also known as I said, deoxyribozymes or DNA enzymes. These DNA molecule can also exhibit or having enzyme activity through the similar structural motif. Now, third point, you have to remember the mechanism, how it will work. So, various mechanism to catalyze chemical reactions by these enzymes. Most well-known mechanism, you know that RNA cleavage reaction catalyzed by the ribozymes. It involves a coordinated attack of nucleophilic groups, where on the phosphate biester bonds or backbone of that, and it requires a substrate RNA molecules that will resulting effect in the cleavage at specific sites. So RNA cleavage reaction that catalyze the hammerhead ribozyme. That words you have to remember. That involves a coordinated attack of nucleophilic group on the phosphodiester backbone of the substrate RNA molecules, resulting in the cleavage at specific site. Now, third point, what are the diversities you will find in the DNA enzymes or ribozymes? So, as discovery or discovered by the wide range of catalytic activities, you have to remember the breakage and joining. So, you know DNA and RNA nucleic acid. Okay. So, RNA cleavage, RNA joining, then DNA cleavage and DNA ligation. Ligation means joining. So RNA ligation, DNA ligation, RNA cleavage, DNA cleavage, and to another major function or discovery of wide range of these catalytic activity involves the RNA phosphorylation and metal binding too, metal ion binding too. So these diversity highlight and the versatility of the nucleic acid as a catalyst, and it has a numerous potential applications. So what are the application now? So as we know that these catalytic nucleic acids or ribozymes or DNA enzymes have a application in various fields of biotechnology. So there are three functions. You can say the application of DNA science. One is gene silencing, another is gene editing, third is biosensing. So three major functions that can use for these three functions. For example, DNA enzymes can be engineered to specific cleavage target RNA molecules, providing potential therapeutic approaches for specific disease. That causes wherever you will find the aberrant RNA expression that causes a disease, and that can also a potential therapeutic use for detecting or you can cleavage also that RNA molecules. Now ribozymes can also be used as a tools for what? For studying the RNA biology, as well as nowadays it work as a potential target for antiviral drugs. As you know that viruses are mostly made up of double-stranded RNA molecules, single-stranded and double-stranded also there. So that can also use for these ribozymes or DNA enzymes can for potential target for antiviral drugs. Now evolutionary significance, how this can helpful in the important implications for the origin of life on the earth. As you know that still today in the molecular biology and biotechnology, they are giving the terminology central dogma. DNA to mRNA, mRNA to protein. So transcription and translations. So the same thing, it is suggested that the RNA molecules have play a crucial role in early biochemical processes before the emergence of protein enzymes. So cannot say that protein to mRNA, mRNA to DNA. So generally they are speaking like this way, DNA to RNA, RNA to protein. Why this is? Because this hypothesis is supported by these ribozymes. Ribozyme then get catalyze involves the RNA replication and metabolism. These words you have to remember. These hypotheses. Which hypothesis? Tell me. Discovery of ribozyme has important implication for what? The origin of life on Earth. So the DNA or RNA molecules that have a crucial roles in the early biochemical processes before the emergence of protein enzymes. So these catalyzed reaction involved in the RNA replication and metabolism. Now structural studies where it is applied. So application you should know. As I said in the previously that DNA enzymes or RNA enzymes having a three-dimensional structure. So three-dimensional catalytic nucleic acid having a three-dimensional structure, and it is elucidated their mechanism or you can engineer them to specific application too. So there are two major instrumental techniques, X-ray crystallography and nuclear magnetic resonance, that determine the structure of various DNA enzymes or ribozymes. Now, last, I will give you a 10 point. First is engineering catalytic nucleic acids. Every words it attached with the previous subsequent. As many researchers have developed a method for engineering these DNA enzymes and ribozymes. Rational design based on, as I see, that structural information or direct evolution approaches. Now, selection or systematic evolution of ligand by exponential enrichment. This selects for docking knowledge. Docking. This technique is very important for the generation of nucleic acid catalysis with enhanced activity or specificity. So ligand binding sites and their specificity. Now what are the challenges? So according to the, when we compare with the protein enzymes, it is very effective. Whereas these DNA or nucleic acid enzymes or DNA enzymes are relatively low catalytic efficiency compared to protein. So it has to be improved for the rational use or direct evolution for specific strategy, as and when we utilize for delivery of nucleic acid catalysis into a cell or tissues, or when we are utilizing for the therapeutic applications which require a efficient delivery system, so that you can this challenge, major challenge, you can retard with the catalytic efficiency, you can improve compared to the protein enzymes. Now, what are the future prospects of these catalytic nucleic acid? So, area of research and many future prospects, as I said in the previous slide. In concluding remarks, you can say that these catalytic nucleic acid or ribozyme or deoenzyme can understanding the structure, mechanism, and engineering. Three words. Principle can likely to lead to the development of novel nucleic acid base catalysts, catalytic with enhanced activity, as well as their specificity. This catalyst could find application in the various fields like molecular medicines, biotechnology, and synthetic biology, or evolutionary biology, or RNA biology, that are larger application nowadays. So this is all about the nucleic catalytic nucleic acids or ribozymes or DNA enzymes. That can also ask is, write down a structure, mechanism, and applications of DNA enzymes or ribozymes. So you have to put it like that. Okay.